While analyzing proteins, often we want to understand results from different tools, but to do that, we need to jump through many web servers, one for each tool. If the wait time in public queues is too long, we need to understand how to install these tools to run them locally, which means learning Python and often debugging Docker files. Finally, once the tools are installed, we need to make sure that we have costly GPUs or high-performance CPUs to even run these tools. Frustrated with this painstaking process, we've built Choppr. Choppr is a no-code, completely online protein analysis platform that lets you really analyze and understand proteins. It allows you to perform very intuitive comparisons. It is completely reproducible, so you don't need to go back to your notes to remember which parameters were used because the Choppr graph has all the details. It is highly customizable. You can create any and every graph you can imagine. It is completely online, so you can access it from anywhere. Let me start by logging in. I already have created an account. Now that I've logged in, I can see all the projects that I have. Let's create a new project called Demo Compare and click on Create. Once the project has been created, inside it, we can create a new job. Let's call this job Compare Trees and click on Create. Now that the job has been created, we can see the input node. The input node is where we can upload our protein sequence files. Double-clicking on the input node or right-clicking and then clicking on the info icon opens the input upload panel. Here we can upload a FASTA file. Or, to try out the tool, we can use the Paste Example button. For this demo, we will use a FASTA file from Unipra. Let's choose this protein sequence for insulin and copy the FASTA file. Now let's paste the FASTA file in the Input Upload panel and click on Submit. The status shows uploading input. To create a phylogenetic tree, we will first search similar sequences using NCBI BLAST. Next, we need to align the sequences. We will use the program MAFFT for this. To chain two BLAST and MAFFT, we just need to right-click on BLAST and select the arrow button to create an edge to MAFFT. Finally, we will select FastTree from many tools that Choppr provides to create a phylogenetic tree. It is very easy to modify any tool's parameters. Simply right-click on the tool and select Info button. For this demo, I'll modify the database parameter for BLAST from SwissProt to NR. To save the changes, just click on the Submit button. Finally, we will run the workflow by clicking on the Run button. The color of the node denotes the status of the tool. In this case, input is green because it is done, and BLAST is yellow because it is running. Now that all the tools have finished running, we can see the results of any tool. Let's right-click on the BLAST tool and select Info button to see the results. As you can see, it has searched 50 sequences with all the important information such as E-value, percentage identity, etc. Similarly, for MAFFT, we can see the parameters that were chosen. MAFFT has finished running, so we can't modify the parameters anymore. The MSA viewer shows the alignment of the sequences with many useful features like wrap MSA, etc. Now, let's look at the results of FastTree. The phylogenetic tree is shown in the tree viewer. Now, to compare trees, we can click on BLAST to use the already searched sequences and build a different tree. For this tree, let's use MUSCLE instead of MAFFT to align the sequences. Similarly, let's use PhyML instead of FastTree to build the tree. We don't need to wait for all the tools to finish running. At any time, we can see the results of the tools that have finished running. For example, while PhyML is running, let's analyze the alignment from MUSCLE. Now we can easily compare the phylogenetic trees from both FastTree and PhyML. Choppr is the only platform which allows for complete manipulation of the data. This is done by creating a copy node of any tool that is finished running. The copy node is square in shape and has a dashed incoming edge. As an example, let's create a copy node by clicking on FastTree and selecting Copy from the menu. Let's create a new alignment of a subset of the phylogenetic tree. To do this, we need to open the output of the copy node and simply select a node in the tree, and automatically the children of the subtree are selected. To realign, all we need to do is connect MAFFT to the copy node and click Run. The MSA viewer of this MAFFT node shows the alignment of the 10 sequences we selected. With this demo, you can see how straightforward it is to use Choppr to analyze proteins. We are looking forward to all the graphs you will create with Choppr. Sign up for Choppr, it's free.